In this short video, we'll review basic enzyme kinetics and its relevance to metabolism. To measure basic enzyme kinetics, we choose one concentration of purified enzyme in lots of different tubes and measure the rate of reaction at several different substrate concentrations. So let's imagine here we're going to measure the kinetics of hexokinase. We'll have, say, 1 nanomolar hexokinase in a bunch of different tubes. We'll have the same concentration of ATP in all those tubes, and the substrate we'll be changing is glucose. So we'll have a bunch of different glucose concentrations. That will be the substrate that's changing, and we'll measure the rate of reaction at each of those different substrate concentrations. Here's the rate at the lowest substrate or glucose concentration. 2 more, and a few more until we can see that saturation has been reached. Now we fit these data to the Michaelis-Menten equation shown here. This gives us the Vmax, the maximal rate of the reaction under these conditions. And Vmax is the same thing as Kcat times the enzyme concentration. So Kcat is the turnover number for each molecule of enzyme. That times the amount of enzyme present is the same thing as the Vmax, or the maximal rate of reaction. The Km is the substrate concentration at half the maximal velocity. So here, if we draw the line to half of Vmax, we can determine the Km value for the enzyme and its substrate, in this case hexokinase for its substrate glucose. So Km is the concentration of substrate at half the maximal rate of reaction. It's an approximation of substrate affinity. And remember that the lower the Km value, the higher the affinity for the substrate. Therefore, mutations that increase Km usually will decrease the rate of reaction for a given substrate concentration. Most mutations decrease enzyme activity. This can happen by several mechanisms. One is to decrease the amount of the enzyme present. This will decrease the rate of reaction, or Vmax. Some mutations decrease the activity of the enzyme. This can either happen by decreasing the turnover rate or by increasing the Km. If you'll recall that RAS, the RAS GTPase, its specific mutation that's very common in a variety of cancers is a specific mutation that decreases the turnover rate for GTP hydrolysis, causing RAS to remain in its GTP bound state longer. And don't forget that enzyme deficiencies can be complete or partial. Some of the patient cases we'll be examining, the patient has zero activity of a specific enzyme. In other cases, they still have some remaining activity. There are some enzymes that are absolutely essential for life. Therefore, if we have a living person, we know that they have some activity of the enzyme. So what can go wrong to cause pathology when there's an enzyme deficiency? Well, let's imagine we have a simple metabolic pathway shown here. A is being converted to B, which gets converted to C, which gets converted to D. And each one of these arrows is actually being catalyzed by an enzyme. So let's imagine there's an X, a Y enzyme, and a Z enzyme here. So what happens if we have a deficiency in enzyme X? Well, the first possibility is that metabolite A might build up, and metabolite A might be toxic to cells. Or accumulated metabolite A might be converted to metabolite E, and metabolite E might be toxic to cells. Or maybe this metabolic block prevents sufficient amounts of metabolite D from being produced. For example, D could be glucose, and we end up with hypoglycemia. Or maybe D, or something downstream of D, is ATP, and the cells just aren't producing enough energy. There could also be a combination of 2 or 3 of these mechanisms. This leads us to a basic classification system for enzyme deficiencies or inborn errors of metabolism. There are those that decrease energy metabolism, those that cause a buildup of toxic metabolites, and those that prevent catabolism of complex molecules. This third category is typically seen with the lysosomal storage diseases and peroxisomal diseases. Here are some examples leading to decreased energy metabolism. Here are some that primarily cause buildup of toxic metabolites. And finally, the complex molecule disorders. While most enzyme deficiencies cause a decrease in enzyme activity, there are a very few special known mutations that actually increase enzyme activity. So there are some specific missense mutations, so remember changing a single amino acid, or a promoter mutation that can increase the amount of enzyme, increase enzyme expression, and therefore increase Vmax. There are a few specific missense mutations that are known to increase the turnover rate, or kcat, of an enzyme. And finally, there are a few known specific missense mutations that increase substrate affinity, thereby decreasing the Km. So what could possibly go wrong if there is excess enzyme activity? Well, there could be increased levels of intermediate metabolite B, which could cause increased levels of some metabolite F, which could be toxic. Or there could be increased levels of all of the intermediates, including the final product of the metabolic pathway, and that could result in pathology. Here are a few known examples of mutations that cause excessive enzyme activity. Enzyme mutations usually cause deficiencies that result in decreased energy metabolism or the buildup of toxic metabolites, and sometimes a deficiency can cause both of these things at the same time, or the inability to metabolize complex molecules. And finally, there are a few known rare specific examples of mutations that do activate enzymes.